Greetings, everyone. This is Mathew Cherian, Executive Committee member for International Pharmaceutical Society. I'll be the moderator for this webinar. And Mr. Kaushik Desai will join me from Mumbai, India as the co-moderator. This webinar is on messenger RNA technology for vaccines and therapeutics. As you know, this is the technology that was used by Pfizer in developing the first COVID vaccine. Now, this technology is not limited to vaccines alone. It can be used for therapeutic agents as well. And there are several of these under development in several pharmaceutical companies. First, let's go to the next slide. And the next one, please. Thank you. Okay, let me go through some housekeeping announcements. This entire webinar is being recorded and it's also live streamed, as you know. The recording will be available for you or your colleagues and friends to watch for a whole year at our website. And you can see the URL for the website right here. You are able to ask questions. Please use the box that you see at the bottom of your screen to type your question. Please make the questions brief because we have limited time. After the webinar, you are welcome to provide your comments and opinion, and there is a URL for that as you see here. Last but not least, we welcome you to become members of International Pharmaceutical Society. Next, please. FIP would like to thank BDO for supporting this online event. As you know, this webinar is in live translation into Spanish, Portuguese, and Mandarin. And the cost of translation expenses are borne by BDO, and we are truly thankful to them for that. Next slide, please. So this is our webinar plan today. The next slide. Okay, so these are the learning objectives, and our panelists will go through that in detail. Next slide. Let me introduce our two panelists. These are two thought leaders in messenger RNA science and technology. Dr. Michael McCaman is a process science expert with vast experience in messenger RNA technology. He has led teams developing vaccines, recombinant therapeutic proteins, cell therapies, and a number of other highly specialized research areas. Our second panelist is Dr. Seth Alexander. He's a chemical biologist trained at Yale University. He has over 7 years of experience in the industry in designing and modifying messenger RNA. With that, I'm going to turn this over to our panelists. So Seth and Mike, it's all yours.
All righty. Thank you very much, Matthew. We'd like to express our appreciation to ISPE and to FIP for inviting us to present today. So on the next slide, so Seth and I discussed how to try to approach this and came up with the concept of trying to ask 10 questions that we thought would be very guiding to a newcomer to the area that would hopefully give a fairly complete perspective on the information that's out there. And so we will try to process it for you in that respect. So Seth is going to lead the first half of the presentation, and I'd like to turn it over now to him to explore the science of messenger RNA.
Thanks, Michael, and thanks everybody for your attendance today and for this opportunity to present this to you guys. So I thought I'd start out with the first question around what messenger RNA is, talk a little bit about the structure and chemistry, and we can build upon that to think about how to make a messenger RNA for a therapeutic application. So as I think this talk deserves, it all begins with the central dogma of biology. This is something I think we're all familiar with. The DNA can be transcribed into RNA, RNA can be translated into protein, and that tends to be what is the central dogma. But it's a little bit more interesting than that in the sense that while over 3/4 of the DNA is made into messenger RNA and other types of RNA, including ribosomal, SNO, et cetera, less than 2% of that total RNA content ever makes it into being translated as a protein. So it really highlights the centrality of RNA within these three major domains of biomolecules in terms of the centralness and cornerstone of what builds life, right? And of course, the story is a lot more complicated than just DNA goes to RNA goes to protein. RNA can replicate itself. It can be reverse transcribed into DNA. And even more, exaggeratively, it can interact with DNA. It can interact with proteins. It really stands at the end of the day as the programmable biotechnology that nature uses to build, to regulate, and to control biological processes. So to kind of start in with the chemistry and the foundations, here I'm taking kind of a snipped view of a single nucleotide. So this is the chemical structure of what an RNA looks like. It kind of differs in DNA in the sense that there's this extra 2' hydroxyl group that I'm pointing to here, but otherwise consists of a nitrogenous base and a 5-membered sugar ribose ring. Now, when you think about what that looks like in more of a polymeric form, so it's a sequence of many of these subunits stitched together, you can see what that looks like here is a representation that represents the 4 different nitrogenous bases that are used for messenger RNA and all types of RNA. Now, another key difference between DNA and RNA is that while DNA uses C, G, and A as their primary bases, it would replace U with T. So there is a minor difference there in addition to that 2' hydroxyl. The other thing I want to point out about the chemistry, just for the organic chemist in the audience, as it were, is that there is also a directionality in this. And if we all remember our sophomore organic from undergrad, there's the numbering to this ring structure. And this becomes what is the 2' hydroxyl. So you hear a lot about 2' methylations and such when you're talking about nucleic acids. This is a 3' carbon, which is one end of this strand, and a 5', which is the other end. And so the strand now has a directionality. So when you hear 5' to 3', it really comes off of the directionality of this backbone that's formed of these phosphodiester bonds along these sugar rings. Now, specific to messenger RNA has a 5' cap structure. And this is a representation of what one of those caps can look like. Most key and notable is this 5' triphosphate linkage, which is quite unique to messenger RNA, that puts on this cap 7-methylguanine cap structure, is highlighted by this green piece here with this methylation. Likewise, there is a 2' methyl group at the first base. This is what differentiates the difference between cap 0 and cap 1. So if you've heard those terms before, that's really what that has to do with. Now, in addition to kind of the canonical bases and cap structures, there are base modifications that even occur naturally. And in fact, tRNA is full of these. Messenger RNA has some of them as well, but kind of taking an eye towards what we've used in the synthetic community for making messenger RNA, these are kind of some of the most well-known base modifications. So a replacement of cytosine with this 5-methyl cytosine. So you can see the cytidine, there's just this single methyl group. Pseudouridine looks very similar to uridine, with just a simple rearrangement of the placement of that heteroatom in this chemical structure. And then of course, N1-methyl pseudouridine, which is the hallmark, I would say, of what's been used in, say, the recent FDA-approved vaccines from Moderna and Pfizer, has this modification structure, which plays some key roles in innate immunity and process development that we'll allude to as we continue the talk. So when we kind of zoom out and look at this, obviously we can't draw the chemistry all the time. And if we zoom out and look at this in terms of what the design features that are an mRNA versus another type of RNA, there's some key features that I think pop out very quickly. So at the 5' end, there is this 5' cap structure that I alluded to on the previous slide. And this is really important for translation initiation. This recruits ribosomes. It also has many different methylation variants, as I alluded to, the difference between cap1 and cap0 that play roles in terms of the stability and even the innate immune response recognition of such elements. Now, as you kind of travel along this mRNA, you see that there's usually a number of bases that are not translated into any sort of protein. This is a 5' untranslated region. It's containing various regulatory elements, and can modulate things like translation rate and stability, and in some cases can even recruit ribosomes itself. Moving on to the meat of the messenger RNA is the open reading frame. This really encodes for the target protein. And if you think about what actually builds this RNA, and builds this protein, I should say, is a codon, and a codon is comprised of three different bases that encode for one amino acid. So if you think about 4 bases with 3 at a time, that's 64 different combinations, but yet only 20 amino acids are typically encoded. So there's many different codons that speak to the same amino acid. So that provides a wide breadth, very wide breadth of sequence diversity that can end up making the same protein at the end of the day. Now, the 3' untranslated region, again, more sequence. Eventually, you hit a stop codon, and there's going to continue to be more sequence in this mRNA, which could have to do, again, with more regulatory elements, modulating the translation rate. But finally, at the end, you arrive at this 3' poly A tail. And this length can vary from low, even sub 100, but usually around 100 is pretty average, up to thousands. And this can affect things like stability, and the translation of the message in a lot of ways. And it provides that stability on the 3' end in a very similar way to the 5' end cap provides stability from exonucleases there. So, kind of moving on to what the biology of this mRNA is in a natural life cycle. So I think it's important to know how mRNA functions natively in your cells before you think about how to hijack it for therapeutic applications. So this is a nice figure that I adapted from the literature that I think highlights some of that basic life cycle understanding of the metabolism of messenger RNA. So mRNA is made of the nucleus, of course, being transcribed out of the DNA in your nucleus. And there's several different processing steps that are undertaken here. There's various modifications of those bases sometimes that can happen. There's a 5' capping process that puts that cap on. There's obviously splicing things that occur. So if you've heard of introns and exons, this is the ability of taking that full length DNA, make it into RNA, you splice specific pieces out and stitch them back together. And finally, it's also polyadenylated to add that poly A tail. And this is what we then call what might be a mature mRNA that's then ready for export into the cytosol, where it can start engaging with the function that it's intended to do. And there's also a lot of proteins that chaperone all of these processes. So an mRNA, when it enters the cytosol, is very highly decorated and very regulated and controlled. And more or less, when it enters the cytosol, it's at this play between 2 different pathways. In one way, you're looking at degradation pathways. So you can decap the 5' end, which makes that end susceptible to degradation. You can deadenylate the 3' end, which would make that susceptible to degradation. There's also uradenylation that can have degradation pathways. And those are basically in direct competition with recruiting ribosomes, usually through cap binding proteins. And we'll go through that in a little bit more detail on the next slide, that actually recruit these ribosomes to start making the protein of interest. And so all this occurs in the cytoplasm. So you can imagine when you're delivering a synthetic RNA that maybe doesn't have all these process controls and decorations, there are some differences that are going to pop out, right? And we'll go through those in details on the next slides. So kind of zooming in a little bit on the ribosomal engagement, the actual function of the messenger RNA. Here I'm borrowing from a figure of the literature that highlights the most canonical method for recruiting ribosomes and is really responsible for the translation and the rate limiting step of the translation process of a messenger RNA is this recruitment of these EIF4 proteins that come together, recruit the small ribosomal subunit, and that ribosome subunit starts scanning along that 5' UTR until it encounters a start codon, in which case, and again, this is a very simplified view, there's a lot of other factors that aren't being shown here. But in essence, when you hit the start codon, the large ribosomal subunits recruited and translation initiation can begin. And that's the most canonical method that this occurs. Now, there's many other methods. Here I'm highlighting one, but of several other alternative translational ribosomal recruitment mechanisms. In this case, an iris is a very common one. This is a ribosomal entry site that can be encoded within that 5' UTR sequence. And it's really this aptameric structure that can recruit these small ribosomal subunits without the need for a cap and then initiate translation in a very similar fashion, right? So multiple ways to skin the cat here. And I think thinking about how you design these mRNAs from a therapeutic perspective and how you use these various pathways is really important in thinking about mRNA design. Now, while this may be the rate limiting step in terms of initiation of translation for ribosomal recruitment, once these ribosomes are on the main open reading frame, they begin translating. And you can think about this a little bit like a one-lane highway. And you have all these cars or ribosomes on the road that are driving along, trying to make their protein. And they can do things like bump into each other and cause collisions and have literally traffic jams. And this can have a large impact on the amount of protein that ends up getting produced. And in fact, there's been a lot of stuff studied about how this exactly happens and how you can modulate that. And if you remember, I spoke to the fact that multiple different sequences can encode the same protein. Those different sequences can modulate the amount of these type of collisions that may occur. And this has been highlighted in some really nice work and from several different authors. And I don't mean to go into too much of a excruciating detail, but just to kind of show you the variability of translation rate and protein synthesis rates that happen as a result of these various ribosomal engagements and abortive terminations and traffic jams that occur. So you really want to keep the ribosomes gliding along the mRNA, not bumping into each other to maximize your protein translation. So that's kind of how an mRNA makes a protein in a nutshell, but that's the biological pathway. So what are kind of the differences and what's different about it when you're thinking about designing a messenger RNA and implementing it in a therapeutic context, whether that be vaccines or others, as we'll come to talk about. So this is kind of a text-heavy slide. I'll apologize for that, but I think communicates and sets the stage as to what's different. So the mRNA is made synthetically. It doesn't go through nuclear processing. So you're not talking about splicing. You're not talking about all these regional modifications, associated protein chaperones, et cetera, in the cytosol. So when you deliver a therapeutic RNA, it already looks very different to the cell than an mRNA that might be made by the cell itself, right? Of course, the goal is still the same. You want to harness the patient's host machinery to manufacture these proteins. And because mRNA is meant to be the immediate intermediary messenger, it's not around very long. It's meant to degrade pretty rapidly. And so it kind of has this built-in off switch, which makes it really useful for applications like vaccines, as we've come to see, as well as things like gene and cell therapies, where you need a protein to be expressed for a short duration to do some intended function and then is no longer necessary. And so mRNAs really are a sweet spot for those applications, hands down, right? Now, again, because these mRNAs are quite unstable. They don't mean to live around very long, and the environment in your blood has many different nucleases that exist. You need to think a lot about that formulation, and it's not just a formulation like you would think of in a traditional sense. There's really, it's providing some key attributes to the mRNA to make it efficacious. For one, it provides stability. Right? It's providing stability of the RNA from those environments until it gets to its target cell. And two, it has to be able to deliver that mRNA to the appropriate cells and even make sure that they get entered into the cytoplasm. And we'll go through that in some later slides in a little bit more detail. So, while that's kind of the overarching overview, I mentioned before that these RNAs look very different. And so avoiding the innate immune response is really important. Biology has really set us up to realize that we've had a long history of RNA and DNA viruses attacking organisms since life began, right? And so naturally, all higher order species have built pathways that stop foreign invading nucleic acids from viruses, whether they be RNA or DNA viruses, to be able to enter and take over the cell. And so these are some innate immune pathways that we have to be conscious of. Highlighted very nicely in a recent ACS Central Sci paper from the NIH, I think kind of highlights some of these specific pathways as important to vaccines. It's a really nice read, I recommend it. But there's things like double-stranded RNA, uncapped RNA that has 5' triphosphates, and most importantly, this TLR toll-like receptors recognize specific domains that promote innate immune responses. And so one reason why N1-methyl pseudo-U was found to be so useful in the pandemic was the fact that there's this additional methyl group that actually prevents this modified U from engaging with the TLR receptor. And so it actually avoids that innate immunity altogether. This was recently highlighted, I think, last year by a nice paper from Moderna that showcased not just the importance of modifications, but even the importance of the process of how you make the mRNA can have a drastic impact on how you avoid this innate immune system, right? So I won't go into these figures in too much detail, except to say that they were showing both U and a modified U, and compared two different processes. And you can see that the double-stranded content can vary widely based upon what process is used. This can, of course, lead to various different IFN beta, which is a signaling marker of the innate immune system. And that, of course, translates to expression. In the case of, say, process B with N1-methyl pseudo-U is extremely immunoquiet and is actually producing the most protein in a reporter assay example. So that kind of gives you a sense of the play of what you're up against as a challenge of engineering these mRNAs to bypass all of this biology that's really intended to keep us from being able to do this, because viruses have been doing it for a while. So I'll take a short pause here and kind of, let another slightly text-heavy slide sink in. And really, this is meant for those who want to review this deck later in terms of key terms for messenger RNA. We've talked about a few of them, like mRNA and cap and tail. But as we continue to go on, we'll talk about things like a T7 polymerase, which is used as one of many possible enzymes that can be used to make an RNA. pDNA or plasmid DNA that can be the template source, and so on and so forth. So a nice reference handy slide here for getting familiar with the lingo, as it were, of mRNA. So kind of moving on to the next question, which is, if that's what you're trying to accomplish, well, how do you actually, at the end of the day, make that mRNA so that you can accomplish all those tasks that I talked about in the last three questions, right? So ultimately, this is done in a cell-free manner now. We've isolated key polymerases and all the components, and we can make RNA from a DNA template source very efficiently. So you take a DNA template. Typically, it's de-ended or linearized. That becomes important, as I'll point out in a moment. But you incubate a polymerase with some buffer, some building blocks called NTPs. These are the individual subunits that are going in. A capping analog is optional. You can actually incorporate the cap right into the synthesis. You can also do it in another fashion. We'll talk about that on the next slide. And then some other enzymes like pyrophosphatase and RNase inhibitor that improve the quality and the efficiency of these reactions. So the RNA, of course, will bind a targeted promoter region on the DNA template that you've incorporated. It'll start scanning and making its RNA copy along. Again, incorporating the cap right from the beginning if you go with the co-transcriptional capping mechanism. And then, I spoke to that linearization. The RNA polymerase and its elongating RNA chain will just fall off the end of the DNA. And if you have an encoded tail and you have a co-transcriptional capping mechanism at play here, you literally end up at the end of a single enzymatic process, a mature mRNA that just needs to be cleaned up in terms of processing. So obviously you add things like a DNase to remove the DNA template or degrade that so that it's removable by purification, and then some downstream purification metrics. And we'll cover those in a moment, but I kind of want to harp in a little bit now on the difference between how you can cap and tail. So as always in biology, there's more than one way to skin a cat. And the way biology, of course, does this, as we alluded to in earlier slides, is you make an RNA transcript that has this 5' triphosphate head on it, and you can incorporate a guanylyl transferase and various different 2' methyltransferases. So this is the vaccinia capping system. It's a very nice protein system that's been harvested from viruses to efficiently put on this 5' cap and get you all the way to a cap1 structure at the end of the day through a couple of simple enzymatic reactions. Now, these are separate steps. They require separate purifications in some cases, so it does add a bit of extra work, but it's one way that you can efficiently cap your mRNA to a cap1 structure. Now, you can also do this co-transcriptionally, as I mentioned. T7 is quite promiscuous in the way it incorporates capping or starting analogs. And so the most canonical and well-known is ARCA. And it more or less provides a cap0-like structure right in from the beginning. Now, that's not always 100% efficient because it's competing with initiation just from a G, for example, if you're using T7. So you're going to have some population, again, of uncapped RNA that you're going to have to deal with at some point. But at the end of the day, you can still get to a cap1 structure very similar to a cap1 structure using secondary 2' methyltransferases, or there are even next generational capping analogs that can do this in one fell swoop. On the other end, there's multiple ways to tail the messenger RNA. So you can encode it and within the DNA, and that's quite nice because it makes a discrete uniform poly A tail length. But the microbial retention of this homopolymeric A sequence can be pretty challenging, especially if you want a longer tail for the bacteria that you would use to make more DNA template through plasmid DNA to be amplified. And so an alternative way to approach this is to use, again, another enzymatic reaction like a poly A polymerase to actually control and elongate homopolymeric A's on the 3' end. And here's some example data that shows how you can control that to some degree, but you're never going to get the nucleotide resolution that you can with a encoded tail. So there's some trade-offs when you're thinking about how you want to build that process, right? So you've made your RNA, but now, of course, you need to purify it, right? So how are you going to do that? Well, the most canonical and academic ways that have been around for a long time now is using basic salt, alcohol precipitation methods, so centrifugation, right? And kind of a slight elevation from that would be, various isolation methods from kits that have been publicly, and commercially available from various vendors, where you can add your RNA into a specific binding buffer, place that on a little spin column, clean it up, wash it, and then eventually elute your RNA, which is fairly pure, coming out the other end, right? But if you think about scalable processes, there's certain limitations, especially with things like centrifugation, if you're thinking about the scales that Michael will allude to in the future. But you want to think about scalable options, and one kind of easy way to think about cleaning this up is thinking about scalable filtration methods, and whether that's a dead-end filtration or cross-flow or transflow filtration method. RNA is a very large biopolymer, and so it's very big, and it's going to get retained in a lot of various membranes, selective permeable membranes from size exclusion. And so you can retain your RNA while getting rid of all those, at least a large swath of those impurities using things like cross-flow filtration or dead-end filtration. But then, of course, there are other methods that are a little bit more elegant and refined in terms of getting rid of specific impurities. So anion exchange is quite nice. This has been well established in the oligo field, I think, to purify shorter oligos, especially when you're making them synthetically, but it can also be used to make mRNAs and purify them. So messenger RNA for GFP, for example, in this exemplary data from a recent scientific reports paper. Now, I think the newest game in town, as it were, is using oligo-DT capture. So you can imagine you can append a small short oligonucleotide of DNA that encodes for multiple Ts, and because a messenger RNA has a poly A tail, that will actually hybridize, and so you can capture if you co-transcriptionally incorporate the poly A tail, say by including it in the template, you can capture only full-length messenger RNA using an oligo-DT type resin, where you can wash all those other impurities away and then elute your mRNA or full-length mRNA product peak that's fully tailed, kind of as its own fraction that gets it away from a lot of other lot of other impurities. So those are a few different choices that one can take when one's thinking about purifying the messenger RNA for the drug substance. So what does this look like when you put it all together for a drug substance, manufacturing train, right? So, typically you're starting with a plasmid DNA source that's coming out of bacteria that's not shown here, but you're going to have to undergo that linearization step so that the RNA has a discrete 3' end by the polymerase falling off. That's going to be incorporated into an assembly reaction that's making the RNA itself. And of course, you have to do that DNA digestion we talked about. And then from there, I think it becomes a little bit unique. And so this is one example with using post-transcriptional capping. But again, there's many different choices, as I alluded to earlier, that one could take. But cleaning it up with, say, some TFF, followed by maybe some affinity chromatography step. And in the case of what's shown here, we may not incorporate the cap until an enzymatic process later. And so you can then put it through an enzymatic capping reaction and then further clean that up to have your final filtration and your drug substance ready to go for formulation. So that's how you make an mRNA synthetically, but how do you characterize it, right? And as I alluded to before, there are a lot of specific features to an mRNA in terms of structure and components that have to be characterized. But at the end of the day, you're really, you're wanting to do things like identify purity, identify identity, and identify any sort of, process or other impurities that may be coming through. So here I'm showing as a figure on the right, using a capillary gel electrophoresis system from Agilent, fragment analyzer that does, I think, quite a nice job of showing the sizing and to some degree the purity of a messenger RNA. Sequence validation is typically handled in the plasmid DNA end, so you can sequence that template, which is transcribed with high fidelity, but there is also ways to, of course, sequence the RNA directly. And then you're thinking about how you're going to quantitate all those process impurities that we talked about. So you've got the DNA template that has to be digested. You have a swath of different enzymes. You have multiple different small molecules and degraded pieces that have to get purified away from. You want to think about, the concentration. How are you going to measure how much RNA you have? That can typically be done by UV absorbance, although there's also other methods that can be employed here as well. And then feature-wise, you have a 5' cap that you need to verify is intact. So there's multiple different ways to do just that. 3' tail as well needs to be characterized. What is your tail length? What does it look like? And then finally, some other impurities like double-stranded RNA, which we know can be quite immunogenic, as I alluded to before. Those all kind of have to be addressed and quantified in various different other assays. Finally, of course, I think at the end of the day, the mRNA is making a protein. And so the biological potency becomes really important to think about the activity of the mRNA at the end of the day. And so building an assay around that is really important for thinking about release of your drug substance, right? So what does a typical panel look like in kind of a phase appropriate manner for when you're thinking about a therapeutic? So here we're covering the basics of purity and identity and safety at the end of the day, right? And then in addition, some of those other features like the cap and the tail and biopotency can be really important, but not all of those are needed necessarily when you're going through drug candidate screening and nomination, right? So when you're doing in vitro screening or even in vivo validation, you really only need a small subset of these to accomplish that. Make sure that you have what you think you do, that the identity is right, that it's fairly pure, that the cap is there, but really, more of these safety and residual enzymes and things really become more important when you're thinking about that IND enabling package towards an NHP tox lot and onwards to clinical manufacturing, where, obviously you want to cover all of these very nicely, right? So of course, that's how you make it, but this is a very fragile, this is a very temperamental molecule. So the delivery is a big challenge for messenger RNA. How do you get this giant polyanionic polymer into the cell that the cell has developed many different defenses to ever get, a foreign piece of nucleic acid into to be used. So to do that, I think most traditionally people have been using lipid nanoparticles. So, I won't go into the LNP side in too much detail because we wanted to focus on the mRNA half of for this talk. But I think a little bit of discussion around delivery, because it is such an important component, is warranted. So, these are self-assembled naturally forming particles that arise by mixing a variety of different lipids as well as other excipients that come together that surround the nucleic acid and encapsulate it into a nice, particle. And it's really doing, again, two functions. It's making sure that you have very nice stability of your nucleic acid, and that it's able to target that RNA to the cell type of interest. And so kind of showing that biological pathway here, if you're thinking about systemic delivery, when you inject this encapsulated mRNA into the blood, for example, for systemic delivery, the RNA would normally be immediately degraded by serum endonucleases, right? And so you want to make sure that the LNP is imparting the stability so that it can get where it needs to go, and then it also has to be able to navigate and get into the cell, and that's done, we think, most traditionally by an endocytotic pathway. So these lipid nanoparticles will bind the surface of membranes and get invaginated in the endosomes. And as that endosome matures, you want the LNP to actually release that cargo. And that's done by a really nice ionizable lipid that can balance that pH. And to kind of give you an idea of what I mean by that, this is a very nice older paper from Peter Cullis's lab, kind of thought as one of the godfathers of lipid nanoparticles. Where you have your LNP encapsulated, and this is of course shown for siRNA, but for mRNA it's, I think, very similar. You have this pH in endosome, so you want as the endosome continues to acidify, you want the RNA to be able to escape, and so you want that pKa of that lipid to be not too acidic, so that as the endosome matures, the RNA can escape. But you want the pKa of that lipid to be acidic enough that it's not degrading when it just enters, the bloodstream at pH 7, right? So, there's kind of a sweet spot for at least liver delivery of siRNA, as shown in this slide, for that pKa to get efficient delivery of the RNA into the cell so that it can engage with the ribosome and make the protein of interest. So what does a process flow diagram look like for LNP encapsulation? Well, in essence, you're taking your lipid components and you're taking your RNA components and you're mixing them together in some sort of chaotic but controlled fashion so that you can have these self-assembly particles to be made in a very producible and reproducible manner. And of course, there's machines that can do this very nicely now, whether it's a simple tea mixing or some more elaborate things that are out by other companies. At the end of the day, you're getting chaotic, efficient mixing so that the RNA can complex with these lipids and form these nanoparticles. Once they're made, you of course have to do some basic cleanup. Again, TFF is pretty traditional to do at least basic cleanup of these lipid nanoparticles and go through final filtration process so that you now have a drug product ready to go to a patient. So what does that typical release panel look like for lipid nanoparticles? Well, again, it's very similar. You're thinking about safety, and you're thinking about identity and purity, right? So LNP sizing, what that distribution looks like, the encapsulation efficiency, how well is it protecting the cargo? How well is it stabilizing the RNA from degrading itself? Is the ratio that you put in the same ratio you get out in your final product? You need to characterize. That's not always going to be the same, right? And then, of course, you want to make sure that you're not producing, some sort of bioburden endotoxin and the safety aspects. And at the end of the day, asking the question around potency of your drug product. Again, not important for all stages. Obviously, in vitro and in vivo, you kind of need to do the minimal amount to make sure that you're making a particle reproducibly, but you're not necessarily as concerned about things that you might be in preparation for IND enabling studies like a tox campaign and non-human primates or on into the clinical stage. So this kind of walks through from beginning to end how you think about what an RNA is, how it goes through to be made, and how it gets formulated. And now Michael's going to take us through thinking about the scalability aspects and a little bit more targeted to the manufacturing and what it's like in the field today. So Michael, I'll pass it to you with the next slide here.
All righty, thank you very much, Seth. That was a great introduction. All right, so let's pick up with question number 7 and start to address what are the challenges to scaling up. So if we go to the next slide. One of the things that's important here on this table is to identify who's actually involved in doing this. and what is it that they need to do. And so what we have identified here are the leading companies that were involved in the COVID vaccine manufacturing, which means these companies were all asking the questions, how do I scale up mRNA? And the answer clearly is it wasn't just one company, there were many. And several of these companies have employed contract manufacturing organizations, or CDMOs, to be able to bolster the total amounts being made, because they were enormous. So it's a whole army of groups that have been mobilized to do this. On the right side of the table, I want to make sure that we have identified that there are different doses of mRNA for each of the different vaccines. And obviously, depending on the dose, from a just simple math point of view, will have a great impact in terms of the scale that one needs to achieve. What I think is remarkable about this entire collection is thinking that this had never been done before. mRNA has always been, up to this point, sort of a low-key research, exotic field to pursue, and suddenly it's become incredibly mainstream, with perceived as the survival of the world, depending on it. So it's a really remarkable story to see how this is worked out. So if we go on to the next slide, let's start to work our way through a little. It's not quite the complex science that Seth was giving, but a little bit of the numerical appreciation of what this really means. So at the research scale on the left side, making mRNA was typically done in several milligram batches. You could do this in a small test tubes. You would order the enzymes that you needed, again, in these sort of Eppendorf tubes, as you see here. And even the purification could be done in little spin columns that are pictured on the bottom. And this would certainly be enough to keep many of the mRNA researchers happily progressing. Fast forward to COVID, and suddenly we're on the right side of this slide and asking questions like, how do we scale up to a pandemic scale that I've tagged this as? So one of the numbers to keep in mind is, and we'll go through this in just a second, is 20 kilograms of mRNA would be needed to deal with a pandemic. By conventional methods, that represents about a 4,000 liter volume of mRNA solution, but that could be made, if being made sort of batch-wise, at a batch per week, you could actually do this in an 80-liter vessel. And so from that point of view, as I've pictured on the right side, an 80-liter vessel is easily done. It's used routinely within the biotech industry. So from a hardware or technology point of view, this is not difficult. What is challenging is in right below that I show, the problem is that the enzymes and the amount of specialty nucleotides that are needed are huge. So if we're going from the Eppendorf tubes on the left to literally multiple Home Depot-sized buckets of these reagents, that's a huge ask of the enzyme and nucleotide suppliers. And obviously they need time to scale up to a level that had never been seen before. So that was quite interesting to watch that unfold. And then the downstream steps that Seth was talking about, again, from the spin columns on the left, we can easily migrate to the right side, where you've got large chromatography columns pictured in one of the panels, and scalable TFF and cassette systems, and they can be built into skids on the right. So again, the idea is that the technology is there to do this, the scale necessary. It's the materials that were really challenging here. Now, if we bring in a center panel, I just want to call your attention to this scale up again. And from my point of view, this is just stunning. If we use the Moderna dose at 100 micrograms per person, injected twice, so that means that for mRNA demands, the 1 milligram serves about 5 people, multiply by 1,000, then 1 gram serves about 5,000 people, multiply by 1,000 again, you're up to a kilogram to serve 5 million people. And if you're going for 100 million people, you're looking at 20 kilograms. That's where that number came from in the upper right corner. If you're going for 8 billion people, well, that's a whole lot of mRNA. But even just going from the 5 milligrams that we had on the right side for a research batch up to the 20 kilograms is a 4 million-fold scale up. Million. That's just, again, stunning that we've been able to, that the science has been able to do this. Just a little footnote, Moderna just shipped their 100 millionth dose. That was a news item this week. So this 20 kilograms for 100 million people is right exactly where the market needs have been. All right, if we go on to the next slide, let's continue the discussion of needs for mRNA, but let's migrate beyond just vaccines. So in this little table here, the Moderna needs were summarized in the top line. Again, to keep that in perspective, what I want to highlight then is below it is that the liver editing or the CRISPR-Cas9 kind of gene editing applications are coming into view now. And as an example, for again, the Moderna dose was like 200 micrograms or 0.2 milligrams per patient. For liver editing, based on some information that I'll provide in a press releases in just a minute, we're looking at a human dose of 21 milligrams. So already for a different type of therapeutic application, you're using 100 times more mRNA. Now, for this particular situation, the patient population is much reduced because these are people with a specific liver disease. So we're using as a model number 50,000 patients. So the 50,000 patients at 21 mg each means you're looking at a 200-gram batch of mRNA to reach that population pool. And this is only, he says, a 40,000-fold scale-up factor. Still pretty darn substantial. If we pull in the little visual here on the right, I just want to put this in perspective, especially the Moderna one. If you look at the cup of water here, or in this case, the cup of tea, there are 2.5 million cups in an Olympic-sized swimming pool. So that's what a million-fold scale-up looks like. So think about that next time you offer a cup of tea to somebody, that if you had to do that to over 2 million friends and followers, that would require a fair amount of logistics and engineering to do that. So I tip our hats to the Modernas and the Pfizers of the world. Just continuing for hold on, we'll finish in the bottom of that slide. Just to continue here, so the liver editing or the gene editing demands, I think you will see grow substantially because that 50,000 patients that we had for an example disease could easily become 500,000 patients for a number of other treatable diseases. And if anybody uses a higher dose, then between these, easily you're going to get into multiple kilograms that are going to be needed for a particular disease. And so eventually, I think what you're going to see is that the gene editing field will certainly keep mRNA demand in the multi-kilogram scale. All right, now we can move on to the next one, please. All right, so now we're ready for our next question. Just reviewing what were some of the challenges in the manufacturing of the vaccine itself. So in the next slide, let's just do a quick review here. This is a sort of an artist rendition on the right side, as Seth has shown in a previous slide, perhaps a slightly more accurate reflection of what we think the structure is. But basically, this lipid nanoparticle is designed to encapsulate the mRNA. It's done, the recipe that I've sort of called out here typically is done with 4 lipids. This could be cholesterol, a phosphatidylcholine derivative, typically a pegylated small lipid, which will sort of create a shell of PEG groups around the LNP that are protected. And most importantly, there's got to be an ionic lipid with a nitrogen in it, so the N-containing ionic lipid. The first 3 lipids that I mentioned are certainly available sort of off-the-shelf materials. The ionic lipid is a very custom synthesis, and there are not that many of them available at different types, and not so many people who can make them. That makes for competition, and certainly in the case of a situation like this, demand, again, a very tremendous logistics challenge. Just to give you a perspective on that, typically one uses about 20 times the amount of lipid as you do mRNA in the LNP production process. So if you're making 20 kilograms of mRNA, you're going to need at least 400 kilograms of lipid in order to encapsulate that all. And while 400 kilograms is not beyond the chemical manufacturing capability of the world, these custom lipids have not been made at that scale before. So again, it becomes a logistics and supply chain nightmare. All right, let's go on to the next slide. For the chemists in the audience, I just want to make sure that we threw a few structures up there so that you can see the different flavors ionic lipids that are out there. We've identified the one that the Moderna is using, the one that the BioNtech Pfizer are using that they've licensed from Acuitas. And you'll see that there are certain similarities in the structures. And certainly the nitrogens that are in there are in slightly different environments, which gives them the opportunity for slightly different pKa's that are listed on the right side. And as that had pointed out back on slide 32, that pKa value has a lot to do with the ionizing properties and the efficiencies of the dissolution of the LNP to release the mRNA. All right, let's go to the next slide. Let's do a little walkthrough of the journey of one of the vaccine vials then, after we've now produced it and vialed an LNP. We start with the picture sort of mid-upper or mid-left there. There's a nice little Moderna vial. It goes in a cardboard package of some sort, obviously. And then we go into, on the bottom left, a shipping container. This is from a Pfizer article that gives you a sense of the effort that goes into, a specially constructed container, lots of isolation with styrofoam, special trays. And obviously you've got to then pack this with enough refrigerant, or in this case, dry ice to keep the temperature during shipping. You'll see over here in the middle bottom panel, the gentleman pouring the dry ice in. Obviously it's cold and requires certain precautions. I think one of the challenges that's probably not appreciated is in that right corner is thinking about the number of vials that you're producing and what are you going to do with them once you've made them and froze them, while you're trying to figure out how to get them from the manufacturing facility to every corner of the country. So freezer farms are definitely the thing that had to be planned for. I think that I would point out, if we give the little animation there, that the lyophilized freeze drying version is on the way. We've heard this from several of the different developers, which could dramatically change this whole supply chain requirement and allow us to either do refrigeration or even room temperature storage for the product, which again would have a huge impact on the ability to deliver for the global needs. All right, on the next one. We wanted to slip this in just for an economic perspective, that this was a little tweet that was shared by the Belgian health minister late last year. And so I put a little translation table in here to give you an idea that what were the companies who were negotiating with the EU asking for in terms of payment rates. And to me, the great news is that these are, if you're putting a human life in balance, these are incredibly, inexpensive materials. What becomes staggering again is the number of patients that need them. But it's nice to know that technology can be deployed and doesn't have to be outrageously expensive. All right, let's move on to question number 9. Let's address something about how else is mRNA being used in the clinic. And we'll slip into the next slide here. So this is a table that I pulled from this review article to give you an idea of the very broad spectrum of viral targets that are currently being actively pursued. Now, mind you, most of these are from Moderna, and kudos to them. And you'll note that they're also staying with the successful platform that they have in play with COVID by using the modified nucleosides as that described for us before. But this, I think, should give us great hope that as a technology, that mRNA vaccines are a real game changer. And to the extent that what they've done for COVID can be deployed amongst this whole spectrum of human diseases, I think is just incredibly encouraging. All right, let's move on to the next one. And continuing to explore a little bit about the non-vaccine needs for mRNA, and in particular focus for the moment on the gene editing. You might remember that in 2020, there was a Nobel Peace Prize awarded to Emile Charpentier and Jennifer Doudna for their foundational work in bringing CRISPR-Cas9 into the sort of the public awareness. And honestly, this is the most impactful science that I've seen in terms of the number of journal publications. The number of scientists who are using this tool is just mind-boggling. We've left the CRISPR acronym over on the left, which you can study on your own if you want. But I think having discovered this editing system and now figuring out how to deploy it is the biggest and most fun element of science that I've ever been involved with. Just as a little footnote, there's a video animation link that I provide below that I think you'll find very educational. That gives you a wonderful artistic version of what gene editing is all about. So if we go to the next slide, I'll do a less artistically ornate version, but try to make sure that we focus on some key points here. The LNP delivers the mRNA with the idea that mRNA content encodes the Cas9 nuclease. It also typically, this LNP for gene editing encodes or encapsulates two small or in a small oligonucleotide or a guide RNA. That guide RNA usually complexes with a Cas9 protein, which is the translation product from the mRNA. And that complex then finds the particular specific gene in the genome, and it makes a double-stranded cleavage. What's interesting is then that the edit function comes not from the delivery of the mRNA, it comes from the host cell's effort to repair that double-stranded break. And it actually is not terribly sophisticated or capable of perfect repair. So it's a misrepair that creates an edit. By misrepair, that means that we're either adding or subtracting an extra nucleotide. So that will shift the open reading frame for any protein that happens to be encoded in that particular segment of DNA. So we call that a knockout mutation. So you've disrupted the open reading frame, you stop production of that protein if it's a toxic or a diseased protein, and that's the therapeutic goal here. If you get a little bit more sophisticated, then in the middle and on the right panels, what you can do is at the same time that you're making the cleavage, you can try to insert or repair that DNA sequence. So if you send in an additional DNA oligo into the repair mix here, the idea would be that by hybridization, the correcting DNA can link and sort of fill in that gap where the Cas9 has created the cleavage, and the DNA sort of self-assembles, and then you've repaired or replaced those nucleotides and fixed the mutation, if it's a single point mutation. If you have to do something more dramatic, then you have to import a greater piece of DNA, and typically adeno-associated virus is being used now for cargoes up to about 5 kilobases. And again, you use the same kind of homology-driven repair to insert now a much larger sequence. And in fact, you can deliver new genes or novel genes. All right, let's move on to the next one. This one is just a quick highlight of a fantastic first proof of concept of in vivo gene editing in the liver. This was Intellia, and Seth and I had the great honor to be a part of this program. And so this has been released. This is some press release statements on both parts, giving you an idea that this is in man. You see on the bottom part that the 0.3 mg per kg dose was a number that we used earlier in the calculation of the non-vaccine mRNA demands. So there's great progress, great news. This whole therapeutic concept is working. It's going to make an incredible difference in the therapy fields. All right, let's move on to the next one. So just to make sure that we give proper attention to the ever-expanding scope of both the diseases and the different editing approaches that can come into play for gene editing that involve mRNA. So I have several companies listed on the left that are pursuing in vivo delivery applications, and several on the right for ex vivo, typically for blood cell modifications. And I've given you company names, and in several cases the particular diseases that they're going after. But I think you'll see this as an ever-growing field and something with great excitement and opportunity. All right, let's move on to our last question here. And so, what is the future of mRNA as a therapeutic? And if we go to the next slide, then we'll paint this picture as being bright and quite fascinating. Most of what we've been discussing today is a linear mRNA, as Seth had described, that it's being capped and tailed in some fashion. And that's where all the COVID vaccines and even the Intellia liver editing have come into play. In the middle panel, I want to point out that there's an interesting variation, which is called a circular mRNA. If you have a circular mRNA, you can imagine that you really don't have the vulnerability that you do with a linear one. You've basically taken away the costs of having to put a cap on the 5' end. You've taken away the cost and efforts of putting any kind of a tail on the 3' end. And by making it circular, you've no longer rendered it open to exonuclease digestion. So it should stabilize. And so that should be a simplified version. That should be a reduction in cost of goods. And all this is driven by the literally internal sequence. So there's a self-splicing intron, sort of a ribosome-like sequence that's incorporated into the nucleic acid cargo that self-catalyzes that circularization. And there are two companies here, Orna and Laurent, who are pursuing that. I think that's interesting. On the right side, I think is an even potentially more powerful one from an economic point of view, and this is called self-amplifying RNA. In this case, the mRNA that's being sent in as the gene of interest, if it's a spike protein from COVID or something, is fine. But now you're using alpha virus replicase genes in that mRNA sequence, and within the cell, those genes are able to actually drive the replication of the mRNA itself. So keep in mind that from before we said mRNA gets amplified in its translation. That is that from one mRNA you can make tens, hundreds, thousands of copies of protein. Now we're saying from each mRNA that's in an LNP or delivered to the cell by some fashion, then now you can make multiple mRNAs. And each one of those will be amplified into multiple copies of protein. So at the very least, the expectation is this will reduce the dose of RNA that would be needed to be therapeutically relevant. So by reducing the dose, perhaps tenfold, maybe more, you'll reduce the cost. You'll reduce the scale of the manufacturing needed to support a particular therapeutic approach. I think that's quite fascinating. All right, and we'll end here with a quick look at the market projections. The panel here that you see is from this article that we cited. It's in a Nature article, and they've given some perspectives that are, I don't know, I'd say perhaps a little conservative in terms of it's a relatively flat revenue curve. What I think is noteworthy is it went from literally zero to a pretty substantial number very quickly. So it's clearly mRNA technology is already profitable, but it's still a young market. The dip that you're seeing there is obviously due to the expectation, one hopes it's true, that COVID demand will be diminished in the very near future. But then let's bring in a panel on the right side from the same article that highlights the directions that mRNA product research is going. And what I want to point out to you more than anything is the light green on the bottom is a huge pool of preclinical work that's going on that is sort of invisible to most of us. So what we're hearing about, what we've seen, is the little 3% that's up above in that yellow square. That's the COVID vaccines. But there's a huge opportunity here, both in viral targets. We've had that table that listed at those. There are cancer targets that are coming on board. The other targets, like the liver editing that we did with Intellia, I think the opportunities here are just enormous. And so what I want to make sure is sort of a going away message here is just for your appreciation that this is a blossoming field. It needs new brains and new hands. Please come join the fun. Be a part of something just fantastic. All right, and I think that'll end our conversation for today. Here's some contact information for Seth and myself, and we can turn it back over to the moderators for a question and answer period. Thank you.
Thank you, Michael and Seth, for a very thorough, comprehensive presentation on a very complex technology. We have a number of questions. I don't know if we will find time to take up all of them, but I'm going to go through them from the first. Here is the first question. There has been mention of using DMSO as a solvent in mRNA lipid delivery. How is this being used and what advantage does it have versus other solvents?
I can say that I'm not familiar with it's being used, so I don't really have an answer to help with in this one. Seth, you?
Yeah, likewise. I mean, I certainly think depending upon, so, and what I'm wondering is if there's maybe, certainly there's a oligonucleotide field that has to do with synthetic oligonucleotides from solid phase synthesis. and that's where you think about a lot of importance of solvent choice. The majority of enzymatic reactions and purification methods that are used for messenger RNA specifically are typically aqueous-based. DMSO, however, being polar aprotic, does provide certain attributes that I could imagine might be useful for thinking about how to keep the RNA and DNA relaxed and denatured and able to transcribe more efficiently. But I'm not personally familiar with DMSO incorporation, so
I don't think it's used in any of the current vaccine manufacturing.
Yeah, no, not that I'm aware of. Exactly.
All right.
Okay, so let's go to the next question. How long, in terms of base pairs, is native mRNA?
Well, that's a good question. It varies widely, right? So if you think about it on the short end, it really varies based upon the size of the protein that you intend to encode and those other elements that I spoke to before around untranslated regions and what gets spliced in and spliced out for native RNA. Again, if you think about it, the number of amino acids, you can multiply that number by 3. That's going to be your absolute complete bare minimum, even too small size for any mRNA. So on the short end, if you're thinking about, some reporter genes that are quite tiny, like erythropoietin, for example, you're well under 1,000 base pairs. On the more extreme end, to the self-amplifying RNAs that Michael alluded to for kind of the future of this, where you're having to encode for multiple other proteins that are enabling manufacture of the daughter mRNA that's producing the therapeutic protein, you can be upwards of 10, 15 kilobases. So quite a wide breadth of sizes on the therapeutic end and also on the native end. mRNAs can have quite a long length, even if they're made by the nucleus.
Okay. Thank you. The next question. Are there translatable eukaryotic mRNAs without poly A tails? Is poly A tail absolutely necessary?
Definitely not absolutely necessary. I think biology never deals in absolutes. And in fact, I think the highlight that Michael spoke to of circular RNA. So this was a recent class of nucleic acids. It's actually naturally occurring as well, coming out of some really pioneering academic labs from like Howard Chang and Dan Anderson and others. I think that you recognize that there are other ways that I spoke to before about recruiting the ribosome. And if you have a closed circle, you may not need for that poly A tail. And so there is a class of naturally occurring circular RNAs in addition to the synthetic ones that are now being capitalized upon in the therapeutic space.
I would also add that you might imagine that the cell would produce a message without a poly A tail if it wants it to be turned over much more quickly. So there's always an opportunity here to customize it within the cell.
Yep.
Okay, the next question. How does messenger RNA induce immune response?
That might take more time than what we have to address. But
yeah, I tried to hit the highlights from the past slide, from a few slides around, TLR receptors, RIG-I stimulation. There's a few other MD5A. I'm trying to remember all the acronyms. It's a little bit of alphabet soup, but what I can say is that there are a number of sequence and modified bases that can change and modulate that innate immunity, as well as process changes and what process residuals and side products are made in a synthetic RNA. They can interfere with those innate immune pathways. But to go into that into much more detail would probably require another hour or longer session.
Okay. The next question. In the event that the messenger RNA doesn't have a uridine modification, how quickly will it get broken down? Will the messenger RNA not be translated at all?
It certainly still will be, right? I think there's not necessarily a requirement for modifications of U. That's just what we found early on, I think, in the field has enabled more efficient translation for synthetic RNAs. But there are other, companies in the clinic successfully that don't require chemical modifications of the nucleic acids. So it's by no means a requirement. I think the modifications simply provide a a boost of being able to be efficient, but you still can make RNA, make protein out of unmodified RNA, and it can be quite effective depending upon the application.
Right, as with all these modifications, the intent is the more that you stabilize the message, the longer it persists and the more translation products you're going to get from it. So again, it's all a question of how much do you need per mRNA to be effective in your dosing.
Okay, here is a question that is based on your slide number 21 regarding the right side of slide 21, namely figure C. Is the implication that the 1-methyl pseudouridine modification doesn't matter nearly as much as using a process that avoids the formation of dsRNA?
I don't know that that's been proven. I think it might have been suspect, but I certainly don't want to end up putting my foot in my mouth. So I don't know that there's a clear answer to that. I think that the modified pseudouridine plays a number of roles, and untangling those differences is quite challenging. Definitely can contribute to multiple different factors, but I don't think exclusively is reducing in any one. But I think Moderna's paper actually showed quite nicely the figure that was on the right of that same slide, that there's actually an impact just of incorporation alone without a process change that can, slightly regulate the changes in double-stranded RNA content. But that's not the whole story for sure.
And keeping in mind that figure represents a single mRNA species, and so different constructs with different underlying sequences might perform a little bit different, even
precisely
in the same experimental.
Precisely. What your application is, what the code is for, those all play factors. So I think it's a little bit reductionist to think about it.
Right. So we would encourage the audience not to take everything that you see here and apply it, across to all mRNAs, but things to be aware of, that there's give and take, there's balances, there's levers to be pulled if you're trying to modulate things. That these are the features to look for.
Next question. What would be the possible factors that would render messenger RNA unable to induce an immune response?
I mean, I might kind of highlight to the past answers a little bit there. I think factors around, double-stranded RNA constant impurities, factors around not efficiently capped material. So 5' triphosphates have a key innate immune activation. This poly-U TLR7 and engagement in terms of how you deliver the RNA can have an impact on innate immunity. So a number of factors, but yeah.
What are the possible adverse effects of messenger RNA vaccine to human host?
Michael, you want to take a first stab at that?
I would say adverse effects would be something that you really want to judge based on clinical studies. But there's no way that there are not effects from anything that you're being given. But relative to an actual COVID infection, don't hesitate to get the vaccine, please, please, please.
Well, then I might also add a little bit of color to say that, mRNA is meant to be a very transient molecule, and what you're delivering is very natural, right? I mean, we made it as synthetic fashion, and it may not be decorated quite the same way, but it is a very similar sister, as it were, of a natural mRNA product. And so it's not around for very long. So it's not like it's staying in your cells forever. It's not doing anything like that. It's making a protein for a little while and going away. So, the mRNA itself certainly, yeah. Exactly.
I was going to say, I think the mRNA is the least objectionable of the materials that you're doing. Obviously, if you're making viral proteins, the way the vaccine is working is you're making a protein that has no inherent effect that we know of, other than to provoke a protective immune response. Keep in mind that the LNP means you're delivering other things too, in terms of these unnatural ionic lipids. So there are other things to keep an eye on in terms of potential adverse effects. That's why you do have clinical studies and you monitor people.
Are the caps and tails vital to prevent immediate degradation?
I think we've kind of addressed that before. And again, the idea is if you want immediate degradation, then ideally don't put a cap and a tail on it. But most people most messages would want some duration, some half-life. So I think that's the trade-off that we've sort of hinted at before.
Messenger RNA is designed not to be very stable, but it provides stability to RNA. Can you explain how does that happen?
Not sure that I follow the question. So you said RNA is inherently unstable and provides stability to mRNA. I'm sorry, repeat.
That's what the question says. I'm not quite sure that I understand that context either.
Yeah, so mRNA I would say is a type, a subtype, as it were. mRNA is a subtype of RNA. They have shared common properties. But if you think about it, the mRNA becomes more stable than maybe other types of RNA once it's engaged with ribosomal machinery, for example. So the recruitment of those ribosomes will inherently block nucleatic activity. But yeah, maybe that's kind of what the question was addressing. But I think, yeah, in essence, it's imparting it through recruiting other functions that keep it busy so it's not being degraded, I think is the short answer.
Is DT resin commercially viable for purification?
I believe so. I'm pretty sure.
Oh, yes.
And yeah,
Definitely.
yeah, like
Yeah.
definitely in multitude of forms as well. Everything from research scale, magnetic beads all the way through, I think some large scale commercial resins, I'm pretty sure are quite available on the market from a number of vendors at this point.
Yes.
What do you think is the biggest challenge in designing messenger RNA for drugs?
That's a good one. It's complicated, right? I don't know that there's any one, but if I had to put my finger on it, I think it's about understanding that broader landscape that we've covered and that you really want to fit for purpose mRNA. So an mRNA that's doing protein replacement is different than an mRNA that's anti-cancer, is different than an mRNA that is gene editing, is different than an mRNA that is vaccinating, right? And this programmable biotechnology of mRNA in general has so many different uses. So I think the application dictates what the most challenging aspect of it is. How do you tune that global process into a fit for purpose that is basically the message for the protein that intends to actually do the therapy in many of the cases, right?
So Matthew, I think there's a question that we skipped that I actually see as could be quite interesting. It was what is the concentration level of double-stranded mRNA that's allowed by regulatory agencies for an mRNA therapeutic? And what is the characteristic differences between single strand and double-strand mRNA that help us to remove this product related impurity? So I think that might deserve a few seconds of comment. So my understanding is there is not a specific regulatory specification that for impurities, typically the agency asks the sponsor to justify what you are proposing to allow to remain in the therapeutic product. So you've got to demonstrate safety. So whatever that number is. Generally, I believe that this is down in single-digit percents. I don't think you want this to be anything substantial, but it's unlikely to be zero. So it may vary from case to case, and may also depend. Again, the question of tolerance may depend on the application. If double-stranded is there and provokes cellular immunity response, and it's part of a vaccine, that might not be a bad thing. Whereas if it was for gene editing in a liver disease where the last thing you want is a huge inflammatory response, it could be that your threshold or your residuals level need to be quite different. So it could, like I said, it may vary for different therapeutic applications, but it could also be argued in a case-by-case manner. In terms of the removal, I don't know that there's the ideal tool is not yet in play as far as I'm aware.
Can we check the bioavailability of the messenger RNA after it is delivered to the body?
That's challenging, but it certainly can be done. I think there's been some really nice studies in animal models. Now, how you would do it in a patient where you can't, cut the tissue out, I think is a challenge. but I would say there's some really nice fluorescent in situ hybridization approaches where you can stain tissues to look for an mRNA over a time course, say for animal models. But definitely, I don't know if Michael, if you've got a thought on how to check it in the actual patient,
Well,
I think is a challenge.
Again, to your point, we know that that's an inherent part of the preclinical toxic studies and demonstrating that in animal models that are relevant to humans. I don't think if this was a situation where you're treating blood cells or something that you could retrieve from blood draw and analyze, that's not too invasive. I don't think too many patients are going to line up and ask for biopsies. And as an invasive procedure, you wouldn't really want to put them at risk. So there are some things that are not going to be easily answered unless there becomes some magic imaging tool, a non-invasive imaging tool that somebody's working on, perhaps.
What is your opinion on the ongoing concerns about the immunogenicity of lipid nanoparticles?
Yes, as I was saying before, for anything that's delivered to the body, you can be concerned about both the short-term and long-term safety concerns, and there's the concern that if you received a certain formulation and you might become sensitized to it, that it might mean that down the road, if after you've had your COVID vaccine, there becomes a gene editing therapy that you're interested in, if you might have some limitations in terms of if you were to have a reaction against that particular LNB formulation. I think we don't know at this point because there aren't people who have been exposed to both, and that's part of the ongoing medical studies that will help guide what's the right path for the different LNPs. And if there's an objectionable one or one that's particularly problematic to people, that'll be the impetus to design better lipids or different lipids to see if we can't reduce those adverse events.
And I might just add one more piece to that, which is to say, in the case of vaccinations, you want to inflame the immune response a little bit for creating an inflammatory response, and to some degree. So there can be times when you want to adjuvant the delivery for immunogenicity. So it's a play of stimulating adaptive over innate, obviously. But lipids can actually be a valuable tool there as well. But I think LNPs are just one way to deliver RNA, and I think I would encourage especially those in the academic community, new ways of delivering mRNA and all nucleic acids for that matter is certainly an active area of research and a field ripe for picking.
How impactful is the messenger RNA vaccine compared to the conventional weakened viral vaccine?
I don't think there's not a version of COVID vaccine that's available as a weakened virus. I think you have the luxury that with a subunit vaccine, the way the mRNA is, so only a portion of the material is presented to the patient. There's no risk of a viral genome getting in and creating any kind of havoc. I mean, obviously, the Salk vaccine solved issues with long ago, but has had its own challenges coming through. But this one, I think the question really is, what's the best subunit vaccine to use? And mRNA is great, low dose, it's effective, it's quick. We were able to respond to the pandemic needs. It's not that there's only one way to vaccinate people. COVID particle peptide vaccines may be coming on right behind us. With people doing synthetic peptides. I mean, there are other ways to do it, certainly.
What is the state of the industry now? Are there CMOs specialized in downstream messenger RNA products manufacturing?
There certainly are CMOs who have been involved with the Moderna and Pfizers of the world, and so that expertise is broadened. Those are CMOs that did not exist, or that function within them did not exist 5 years ago. when I was first looking for it. So yes, they're limited, and some of them are still very much tied up contractually with the ongoing COVID manufacturing production needs. But yes, there is a growing body of CMOs who can work within the mRNA field, from the research and design of mRNA all the way up to the GMP production.
Can polymeric delivery systems be used for delivering mRNA vaccine?
Can adjuvant vaccines be made? Well, in a sense, the LNP is acting a little bit like an adjuvant. Could it be bolstered? Could you get more? Possibly. Do you need that? Don't know. That'll depend on the clinicians who decide to present these. I don't think it's necessary at this point. I think the mRNA ones are showing their ability that with tens or of micrograms of injection, you're getting protective response. So adjuvants are certainly a crucial where it's a very weak immunogenic vaccine, and I don't think that's a problem here.
Do we know how much of the injected RNA is translated into protein antigen?
I do not. I think that's an interesting unknown in each of our own shoulders. That's how our body.
I can provide some color from the general community, not specific at all to the even mRNA delivery for that matter. Although I think I've seen at least one or two papers that kind of study this, but I would say that there's a lot more of a depth of field in the academic realm with siRNA field and delivery, and traditional LNP delivery is looking at something like an escape of about 1% to 2%, I think is what I've seen in the literature. So you can imagine out of what's getting formulated, maybe 1% of that actually gets uptaken into an endosome actually finds its way out. In the case of SI, now, does that translate to messenger RNA? That's a question. Definitely an unknown hard answer, but maybe that provides a little bit of context in terms of areas of improvement, if that makes sense.
Okay, so this will be the last question. There are more questions. We don't have time to take up all of those. So this is the last one that I'm going to ask. Pfizer announced last December about a production shortage due to the key protein stabilizer, which low endotoxin sucrose. Are you seeing the same issues today?
I'm not familiar with the supply shortage of that particular compound, but I think in general, as I was saying before, that between the enzymes and all of the other components going in, that there were sort of massive shortages or demand exceeded supply for everything. So it doesn't surprise me that that kind of thing would have occurred. But I think there clearly have been workarounds for all the other components. Otherwise, Moderna would not have shipped 100 million, and Pfizer would not be right up there with them. So I suspect that that they've identified what those problems were, and those are solvable problems. It just takes a little bit of time and a lot of energy and enthusiasm, and I suspect that those have been applied here.
Okay, so we will close with that. There are more questions. The attendees can correspond with the panelists. Their email addresses were on the presentation. So I want to express my deep gratitude to both of them for presenting such a fabulous presentation. This has been very thorough, comprehensive on a brand new technology, and we are truly thankful to you. Thanks, everyone.
Thank you.
Thanks so much.
Mathew, can you please promote the last slide? Because I think attendees will be happy to see it. Yes, this one. Thank you. I will be sharing the link now. So what I did, I shared in chat box the link and the password. I think that this might be interested to see the collaboration between FIP and Euromed. And on the screen there you can see all the details. So this was all from me. Thank you.